So here we will discuss little bit about receptor regulation. Now basic thing to understand, if a receptor is constantly stimulated, constantly we keep on stimulating, the activity of that receptor decreases. And on the other hand, if a receptor is not stimulated for long periods, like we give antagonist for long periods, then its activity increases. Yeah? So receptors will adapt. So how they adapt we want to see. So starting with the first method, that means when we constantly stimulate the receptor, constant stimulation of receptor, or we can say constant presence of agonist, how it modifies the receptor. So important thing to remember, when we constantly stimulate the receptor, the three things can happen due to which the activity decreases. The first thing is masking of receptor. Masking simply means the receptors which are present on the surface of the membrane, which are present on the membrane, they will go down below the membrane. So they are not present for the agonist to work. So masking of receptors can occur, and this masking can occur immediately because the receptors are present, they go down. Immediate masking can occur, quickly it can happen. Second thing which can happen is down regulation of receptors. Down regulation simply means decrease in number. Number of receptors may decrease. They may decrease by two methods. Either the receptor synthesis stops, more receptors are not synthesized, and second, the receptors which are already present, they may be degraded. So finally the down regulation occurs. It is a relatively slow process. It will take around a week. And the third thing which can happen is it is uncoupling. Uncoupling of signal transduction pathway. To understand this uncoupling, suppose this is a receptor, G protein coupled receptor we assume. So the agonist or the drug bind to this receptor. After binding to this receptor, they will stimulate the G protein. We know G protein consists of three subunits, alpha, beta and gamma, and this G protein is broken, then the action will come. Now when constantly agonist is present, then it is not able to stimulate the G protein. The coupling does not happen. So when G protein coupled receptor is not able to couple with the G protein, obviously the action will stop. Now how this uncoupling happens? What happens is, in the cells we have a protein called as GRK. An enzyme is present. The name is G protein coupled receptor. G is G protein coupled receptor, GPCR. Receptor kinase. G protein coupled receptor kinase. So kinase means phosphorylase. So when the receptor is constantly getting stimulated by this agonist, now G protein receptor kinase will phosphorylate this protein. This protein is phosphorylated. We have GRKs for almost all G protein coupled receptors. In case of beta receptors, if we talk about beta adrenergic receptor, so this G protein coupled receptor kinase is known as BARK, beta adrenoreceptor kinase. So this simply is type of GRK. So this will phosphorylate the receptor. Now when the receptor gets phosphorylated, now it will attract a protein which is called arrestin. So this phosphorylated receptor, it binds to a protein called arrestin. And when it binds to arrestin, now it cannot interact with the G protein. So when it is not able to interact with the G protein, or simply uncoupling happens, now the action will stop. Okay? So this is third mechanism, that means uncoupling. So by three methods, we can decrease the activity of receptor when it is constantly stimulated. One, masking of receptor, means receptor goes down, not available on the membrane. Second, down regulation, means number decreases. And third, uncoupling of the signal transduction mechanism. Clear? Similarly, if we constantly inhibit the receptor, or we can say constantly non-stimulation of receptors, constant presence of antagonist we can say, or constant non-stimulation of receptors, it will do the opposite thing. So same three things will be reversed. That means instead of masking, there will be unmasking. Unmasking means the receptors which are present near about the membrane, below the membrane, sideways, they will come on the membrane. so it can work more. Second, instead of down regulation, we will have upper regulation. More number of receptors are synthesized. Again, upper regulation will require little time. And third, there will be increase in signal transduction. So same number of receptors can stimulate the G proteins more. Okay? So that is constant presence of antagonist or receptor is not stimulated. Like we can take the example. If you remember, there is a receptor present here which is called alpha 2. Alpha 2 is stimulated by clonidine. So when we give clonidine, it stimulates alpha 2 receptor and alpha 2 act like a brake. That means noradrenaline release from the vesicle is inhibited. Brake occurs. So when noradrenaline do not come, so these receptors cannot be stimulated. So in the blood vessels, they are alpha 1. In the heart, they are beta 1. So they are not stimulated. So when they are not stimulated for long periods, what happens? Upper regulation occurs. So the number of receptors increase. Number of receptors increase. So due to increase in number of receptors, whenever noradrenaline come, it can act on large number of receptors. Till the time we keep on giving clonidine, noradrenaline do not come. So no problem occurs. But when we suddenly stop clonidine, now noradrenaline comes. And when this noradrenaline comes, now it will find so many receptors. So the action suddenly increases. Like we know clonidine act as a brake to sympathetic system. So it decreases the blood pressure. So clonidine decreases the blood pressure used as antihypertensive drug. But when we suddenly stop clonidine, the BP shoots up, suddenly increases very much, which is called as rebound hypertension. So this rebound hypertension, it occurs because of increase in activity of receptors because of constant non-stimulation of these receptors. So these are adaptive changes. So if constantly agonist is present, then the receptor is not stimulated. When constantly antagonist is present, then stimulation of receptor increases. Clear? So this is important about the receptor regulation.